Everyone at some point in their life has heard that exercise is good for you. It improves general well-being and can help prevent chronic conditions such as heart disease, diabetes, arthritis, and cancer. It has a tremendous impact on the brain and neurological conditions such as Alzheimer's, Parkinson's, MS, and depression. Nonetheless, despite all these endless efficacious effects, have you ever taken a moment to think about why? Or even more importantly, how this all occurs? This question, with an emphasis on the brain, is the basis of my research and what I strive to further explore. It is well established that muscles release molecules during exercise that contribute to some of these positive effects. However, what is not well understood is whether or how these molecules are able to cross the blood-brain barrier and reach the brain. One hypothesis stems from small nanoparticles with a large role in cell-to-cell communication, called extracellular vesicles. As you can see from the diagram up top, these vesicles are produced by muscle and thought to contain these neuroprotective molecules, which are then transported through the lymph and blood vessels where they ultimately reach the brain and release their cargo. If that sounds a little bit confusing, think of your body as a postal system. Now, if you look at the diagram below, you'll see that the muscle cells act as factories, producing packages or extracellular vesicles with valuable contents. Now let's imagine you wanted to ship a package from here in Kingston all the way to Florida. Except you didn't have to pay tariffs, of course. The blood and lymph vessels act as trucks on roads carrying these packages, where they are received by the blood-brain barrier, which acts as a sort of border customs, screening these packages and making sure that only designated safe ones reach the final location. Once they reach their final stop, they are opened and their contents are put to use. To test the accuracy of this hypothesis, my thesis exercises a never-before-used, genetically modified mouse model with glowing, trackable extracellular vesicles that are only produced by muscle. The distribution of these vesicles after exercise throughout the body will be measured using a variety of analysis strategies. If a substantial number of these vesicles are found in the brain, then this would prove this hypothesis correct and demonstrate the vesicles indeed are able to travel all the way from muscle directly to the brain. This would be a monumental breakthrough in the world of drug and development and could be the key to treating neurological conditions. It could allow vesicles to act as vehicles for many neural therapies, allow for the boosting of exercise, and providing those who are unable to exercise with a way to gain its advantageous effects. Therefore, next time you receive a package, I hope you remember the potential role of extracellular vesicles in your body's own postal system.